Hello, my name is Taylor Brown and I am in Biology 2440 Lab, Section 09. This is my membrane concept map. Alright, to start off, my biggest topic is plasma membrane. Plasma membrane is basically the container of all cells and helps mediate transport, in and out the cell signaling, and just overall protection of the cell. From plasma membrane, we have two different models of the plasma membrane that has been introduced by scientists. The sandwich model, which basically is the first model that was ever introduced, and it looks like a sandwich. It shows that the membrane proteins are on the outside, and the inside is the phospholipid bilayer, which is right here. We'll talk about in a second. And the second most accepted concept is the fluid mosaic model, and that shows that the proteins are just not on the outside of the phospholipid bilayer; they are also integrated within the bilayer. All right, so that's a model of the membrane. Now let's go to actually the way that molecules can slip in and out of a membrane. That is through selective permeability. That means that the membrane is selective to what it lets in and out of its cell, in and out of its barrier, actually. Mostly small molecules and specific ones that can go in and out of that. And talking about small molecules, it comes to osmosis. Osmosis is the transfer of water in and out of a cell from an area of high solute concentration to low solute concentration. And there's three different types of osmosis. Hypertonic, which the water rushes out of the cell. Hypotonic, the water rushes into a cell. And isotonic, the amount of H2O within the cell and without of the cell is the same, where the word iso comes from. So there's no net water movement. All right. Now let's go back up to the plasma membrane and talk about how the plasma membrane is formed. Well, actually, new plasma membrane is formed. So when it is time for new plasma membrane to come, things called vesicles bud off from the endoplasmic reticulum and transport all the way to the plasma membrane in little forms of buds, so the plasma membrane can always keep closed. Detergent is pretty much the only thing that can break the plasma membrane. It's a specific type of molecule that works to break up the plasma membrane and allow the contents of the cell to spill out. All right, now that we've talked about the permeability of the plasma membrane, how it's made and destroyed, and what it looks like, now we're going to go to the actual components of the membrane. Now, the phospholipid bilayer is a hydrophobic tail, which faces the inside of the membrane, and the hydrophilic head, which faces the outside, and the two parts of a sandwich. So say the hydrophilic heads are the bread, both the top layer and the bottom layer, and the hydrophilic tail is the meat and cheese. This way, the phospholipid bilayer is amphithetic, meaning that it has its makeup is hydrophilic, that loves water, and hydrophobic, that hates water. It works both ways, like an amphibian. And the hydrophobic tail is a chain of fatty acids that is linked to a glycerol that's linked to a phosphate that's linked to the hydrophilic head, which is choline, serine. It could be a number of things. Now, the hydrophobic tail being a fatty acid, which we talked about last time, it can be unsaturated, which means that it has at least one double bond and not all the hydrogens that it could possibly have are on the molecule, or saturated, that it has all the possible hydrogens it could have. Okay, that is the makeup of a phospholipid bilayer. Now, branched off from that, we have two things. We have, let's go back, this thing is hard to hold, membrane proteins, membrane lipids, and through those membrane proteins, make membrane channels. We'll talk about the lipids first, because it's kind of the smallest one. Bandit, come here. Okay, the membrane lipids are the lipids that connect to the outside of the phospholipid bilayer. They're either steroids or sterols or glycolipids. Steroids help to keep the kinked portion, which is the unsaturated fatty acid, and the saturated portion from just being a space filler. So it basically, where the kinked portion and the straight portion make a gap, those steroids fill that hole to make the phospholipid bilayer more stable. And glycolipids also play a role in cell signaling, cell structure, things as that, helping bond molecules that are hydrophobic, because lipids obviously like hydrophobic type of bonds. Okay, now let's go to the membrane proteins, which are a very important part of the plasma membrane. The membrane proteins are basically the most abundant way for the cell and everything that goes on inside of the cell to happen. So, there are two types of proteins: integral, which go all the way through the bilayer, and peripheral, which is they are not a part necessarily of the membrane. They can always easily be separated and such. So those two, and there's also different functions of the proteins. So you have the ways they're attached, to which are integral and peripheral, and their functions. You have transporter ones that transport molecules, anchor proteins that anchor specifically to the phospholipid bilayer. You have the receptor proteins that receive information from different cells and other such things, and enzyme proteins that attach to the bilayer that help specific reactions come along. And there is 4 different types of integral proteins: proteoglycan, glycoproteins, monolayer, lipid link. And you have your peripheral proteins. Those are proteins attached to other proteins, such as those proteins that aren't specifically integrated into the membrane. Whoa, the wind. It's kind of bad. Okay, so through those proteins, we have, on the phospholipid bilayer, specific channels. Now, this is the specific way that molecules enter and leave the cell. Now, we have 3 different types of ways. Simple diffusion, which basically means that small molecules can enter and leave the cell based on their concentration gradient anytime they want to. Now, gated and ion channels are different. They're a different case. Ion channels can only accept specific charges based on their electro gradient and their concentration gradient, and so does gated. Now, gated is a little bit different because the gated ion channels only open for a specific reason. Ion channels are basically always open, but can only accept specific charges. Gated ion channels can only accept specific charges, but it's only open and closed at specific times when the channel is mediated by passive transport. And passive transport works through also concentration gradients. Passive transport is a type of transport that works by no energy. There is no energy needed for Bandit. No. These specific molecules to move in and out of the cell. But active transport, there is energy needed for the specific thing that's being transported to move across the gradient. All right. I think that is all. There's my concept map as a whole. I've kind of tied everything together. And that is it. Thank you very much.